Yes, ma'am. Oh, good question. I have to go check my plant physiology sources. I don't remember off the top of my head. I think all plants do, but I'm not 100% sure. Plants. Plants.
Wouldn't it make sense that plants that are more in the cold environment would have ultraviolet way more than their plants were in like Victoria?
I'm sure it would make more sense, but I don't remember that that's restricted to cold environmental plants. And plus, not all flowers do this to come out early in the spring. So whether or not they use it for this purpose, there are some species that do that. All right. Here's another inhibitor, dinitrophenol. This is what we call an uncoupler. An uncoupler uncouples the proton gradient from the ATP synthase. So what does that mean? It allows the protons to go back across the membrane, so they don't need to go through the ATP synthase. So it dissipates the proton gradient. So, hold on. Uncouples the ATP synthase from the proton gradient. So the protons do not need to go back through this protein because the uncoupler lets them go back in. So it dissipates the proton gradient. So while electron transport establishes a proton gradient, the uncoupler undoes that all of that work, lets the protons go back in. No more proton gradient, no ATP synthesis. What does the uncoupler do to electron transport? Well, it negates the effect, but what does it do to the electron transport process itself? Well, it affects ATP synthesis. I'm asking you what happens to electron transport. Nothing. It lets the protons go back in. It doesn't do anything to any of these steps. So electron transport continues, establishes the proton gradient, uncoupler undoes the proton gradient. That's all it does. How many ATPs do you get out of your glucose? Well, you definitely get the 2 from glycolysis. Depending on how much uncoupler you have in here, you may or may not get any out of electron transport. Which is the reason why this was used as a diet pill in the 30s. And it ended up killing a bunch of people, so they took it off the market. And the reason that it killed people wasn't because they had little ATP, it was because their body temperature rose too much. So it was as if they had a really, really high fever. And what does a really, really high fever do to you? It enters your proteins, and so they started dying. So like, oh, okay, not a good idea. Yes, you want to look thin, but you want to die. So now it's considered a toxic substance by EPA. So why would this be a good diet pill? Alright, so if you're only getting 2 ATP out of a glucose molecule, so for all the food that you eat, you don't get that many ATP out of it, and of course your body's still working and it still needs lots of ATP, which means you will break down all of your reserves and get only 2 ATP per glucose molecule, which would mean that you end up losing a lot of fat, and you will eat all the carbs you want and wouldn't get any ATP out of them, and so you just lose all of that energy. The problem is that electron transport is still going full force, and you're not getting any ATP out of it. And as electrons go through here, they release heat. But instead of getting 36 ATPs per glucose or 38 ATPs per glucose, you're getting 2. So you have to break down 19 times as many glucose molecules to get the same number of ATP molecules, which means you have to have 19 times as much electron flow without getting any ATP out of it. And all that extra electron flow means extra heat. That's why your body temperature goes up, which might kill you. So next time there is a diet pill fad, you might want to check the side effects before you jump on the bandwagon. Any questions about the inhibitors? Yes, sir. Yes, sir. So what DNP does, it's an uncoupler, so it allows the protons to dissipate back down their gradient. I'm looking into the mechanism of how that works because that's a little too much chemistry for us. Good question, but I'm not telling you. Yes, sir. No, you already have a gradient. It just makes the protons go down their gradient. Because electron transport establishes the gradient, and DNP lets them come back in, down the gradient. So instead of going through the ATP synthase, dinitrophenol allows them to go back in on their own, separately. Well, not really on their own, but allows them to go back in through a separate mechanism. Yes.
Could the uncouplers potentially at high concentration potentially seize the electron transport chain?
The question is whether uncouplers affect electron transport. All the uncoupler does is allow the protons to go down the gradient.
High enough concentration potentially stop.
The only thing the uncoupler will do is allow the protons to go down the gradient. That has nothing to do with electron transport.
And what about, ATP synthase?
So ATP synthase relies on the proton gradient. So if you don't have a proton gradient, there will be no ATP synthesis. Yes. So the question is, why would there be extra heat? So if the DNP is preventing ATP synthesis by dissipating the proton gradient, then for any glucose molecule, you would be getting 2 ATP from glycolysis, and you wouldn't be getting the other 30-something that you get out of this process. So to maintain the level of ATP that your cells need, you're going to have to break down 18 or 19 times as many glucose molecules. And for each of those glucose molecules, you're going to have electron transport without getting the benefit of ATP synthesis from it. So you're going to have 18 or 19 times as many electrons flowing through this process, even though you're not going to get any ATP out of that. And every time electrons are going from one complex to the next complex, they're releasing a little bit of heat. And now you have 18 times as much heat being released, potentially. Yes, sir. Very good question. It's not a channel. There's a chemical reaction which is a little bit beyond the level of this class. So there are multiple uncouplers, and the way they work is that on one side of the membrane they bind to the proton, and on the other side of the membrane they release the proton, and they're hydrophilic on one side and hydrophobic on the other side, and it's a little complicated. So. The DMP would be the, I'm sorry? What do you mean activates it? This is a biologist injecting this into the mitochondria just to see what it does. No, it's a diet pill. It's a chemical that you're using to release the proton gradient. No, this is not naturally occurring. Yes.
Regarding the electron transport chain, the alternative pathway for the electron, what would be the last complex on that alternative?
So the alternative pathway for the plants, it's right after ubiquinone, so it's before complex 3. So it goes from ubiquinone to another protein here. So it doesn't go to complex 3 or 4. Last step is that alternative oxidase, which is right there. The reason that it's less efficient is because there are fewer steps that can allow it to make to establish a proton gradient. So the proton gradient you make is smaller than if you had used the regular pathway. Alright, so if you consider respiration as a whole, most of the energy is going from glucose to NADH and FADH2, and then from them to the electron transfer chain to establish a proton gradient to then make ATP. Now, there is a little bit of ATP produced in glycolysis and a couple of ATP molecules that come out of the citric acid cycle, but most of the ATP is coming from this. The electrons from NADH and FADH2, electron transport chain, proton gradient, ATP synthesis. So the efficiency about 40%, which you might think, God, that's really not that efficient, but it is more efficient than anything man-made. So you still have to be in awe of nature and that this is a highly efficient process. So now we're going to take a look at a couple of experiments that helped us figure out the importance of the proton gradient and the protein ATP synthase in the process of synthesizing ATP through chemiosmosis. So I'm going to go over the experiments with you, and then I'm going to ask you to consider the questions we always ask. What's the big question? What are the alternative and null hypotheses? The experimental prediction. So here's the first experiment. You have isolated mitochondria. You're not looking at full cells here. So you have an isolated mitochondria. You're going to put it in buffer, pH 9, let it equilibrate. So outside of the mitochondria, inside the mitochondria, inside the matrix, everything is pH 9. Then you're going to take those mitochondria out of the pH 9 buffer and move half of them into pH 7, and move half of them into another tube which is still pH 9. That one is not shown in the diagram. That's your control. You keep them at pH 9, but you still move them to a different tube because the movement process itself has to be standardized. So now you have a pH gradient because you artificially established a pH gradient in half of your mitochondria, and in the other half you did not establish a pH gradient. So half of them have pH 9 inside, but pH 7 outside. Half of them have pH 9 on both sides. And then you look to see whether there's ATP synthesis. So that's the first experiment. The second experiment does not use mitochondria. Uses artificial vesicles. And so you can decide which proteins to put in the membranes of these artificial vesicles. So initially, they put a bacterial proton pump in here, which then pumps protons inside the vesicle against the gradient. So there's a high concentration of protons inside compared to the outside. You provide ADP and inorganic phosphate, nothing happens. There is a proton gradient, but there is no ATP synthesis. You take half of those vesicles and insert the protein ATP synthase. And then look to see whether there's ATP synthesis. And they make ATP. The ones without the ATP synthase have a proton gradient, but they don't make any ATP. So consider both experiments. I'll give you 10 minutes. You figure out the question they're trying to answer, alternative and null hypotheses, experimental prediction.